but proteases which are produced from the bacteria are highly useful and are used in various types of industries. And my main objective is the aim of isolation of characteristic bacillus species from detergent contaminant cells. Detergents are used to remove stains from the dirty clothes or various stained clothes, which the detergents are mainly made up of proteins and enzymes which break proteins or proteases. And bacteria, they produce different kinds of proteases. And I have given basically some examples of bacillus which produce protease. And applications of protease are used in various industries like leather, management of waste, textile industry, and detergent industry. And how the isolation of bacillus species are have done in this methodology. And I collected samples from various dhobi goats of Yadavgiri and Kuyempanagar. The dhobi goats are the places where various cloths and stained cloths are washed with the usage of detergents, and the soil there is basically detergent contaminated. And few species of bacillus thrive and they are grown. They can sustain sustain the detergent and then surviving in the back and in the soil. Isolation and screening of proteolytic bacillus species. The soil samples are serially diluted of appropriate dilutions and were inoculated into LB agar. LB agar is basically the media where where bacillus can survive. It is kind of nutrient to the bacillus, and the plates are split inoculated into the agar plates by split plate method and incubated at 37 degrees for 24 to 48 hours. And various colonies of different distinguished morphological colonies were identified, and they are again transferred into the LB broth for 24 hours. And they are point inoculated on the skim milk agar media. Basically, skim milk agar media is made up of protein which is present in milk. The casein is present in milk. This is basically hydrolyzed by enzymes protease which are present in bacillus. When zone of observance is seen, we can clarify that bacillus species has proteinase enzyme in there which can degrade the casein present in the milk. And quantification and protease activity is done after the isolation of bacillus species. The present study is based on the isolation of proteolytic bacteria from detergent contaminated soil for the future application in protein digestion. As zone of observance is seen on the skim milk agar media plates, and standard graph is done using various different concentration of BCA. Graphical representations of protein content and proteinase activities is presented to you. Basically, the total protein content in cell-free supernatant bacterial cultures from the soil is maximum seen in the 9th, which is one of the sample, which is one of the isolates collected from the soil. And the protease activity is basically seen maximum in the cultures of wheat and D, which I have mentioned. The maximum showed is 37.5 micrograms per mg. Thank you for giving me opportunity to share my observation and about protease producing bacillus species to you.